Good morning and welcome everybody to another webinar of TN2. First, some small minor instructions if you're not so familiar with Zoom. Here you can see how you can set up your screen. You won't be able to see yourself on the webcam, but you can send us questions. And the chat box, as you can see, is disabled, but use the Q&A to send in your questions. And please do, because we really like an interactive session. So it is my pleasure to welcome you all to this webinar, and together with my co-host Anke Dijkstra, who you can also see in the screen, we will host for you this great session today. And something about TN2. TN2 stands for Translational Neuroscience Network, and what we aim to do is bridge the gap between fundamental and clinical research, but also towards industry and other applications to really provide an environment for a lot of interaction. And for this, we have these webinars that we started in COVID periods, but now are also going to continue with those this year. But we're also going to set up our annual in-person meetings again. So at the end, you will see more information on this. The meeting this year will be on synaptic plasticity and is actually really exciting. And later on in the chat, you will get a link how to apply for this session. So this year we'll have 4 events, 3 webinars. This is the first one on artificial intelligence, then one in June and one in September. So spread the word, and it's great to see that you are all here in such great numbers. So now I will hand over to my co-host Anke for more information on today.
So today we have 2 great speakers, Tijms and Arman Eshaghi, who will present their works and how they integrated artificial intelligence in their work. And in the end, we have an opportunity to have a more broad discussion to discuss and intertwine all these different topics on artificial intelligence. So it's my honor to introduce the first speaker of the webinar, Tijms. She's an associate professor at the Alzheimer Center at the Amsterdam University Medical Center in Amsterdam. She first obtained her PhD in Edinburgh, where she developed an analysis technique to describe gray matter alterations using MRI. And after her PhD, she joined the Alzheimer Center, where she now leads her own group to investigate gray matter alterations in early Alzheimer's disease and integrate this with CSF proteomics. So with that, the floor is yours.
Thank you very much for this introduction and welcome everybody. Let me see, I'm going to share my screen. Share. Yep, and I'm so sorry. There's something. Play from the start, right? So I hope that you can see my screen now. All right, so I'm going to talk to you a bit about machine learning in Alzheimer's disease. But before I start to do that, I'd like to introduce you into some of the concepts of Alzheimer's disease. Otherwise, I think you won't be able to follow what big questions are. So Alzheimer's disease causes dementia, and dementia is a term to describe a set of clinical features. So it's a clinical syndrome of difficulties with your memory, visuospatial functioning, and other thinking skills. And currently there's almost 300,000 patients living with dementia in the Netherlands. And Alzheimer's disease is the cause of dementia in most of these cases, about 70%. And here we see the person that the disease was called after. That's Dr. Alois Alzheimer on the left. He described the very first patient now almost 100 years ago. And that was Auguste Day. She got admitted in his psychiatric hospital ward with a lot of symptoms, amongst which memory loss, but there were also language problems and severe behavioral problems. And during her stay there, she deteriorated really quickly. And Dr. Alzheimer was just fascinated by all the symptoms that he saw and that she did not improve. So he thought there must be a biological explanation that for these symptoms. So after she died, he decided to look into her brain to find causes of her disease. And what he did, and when he looked into the microscope, what he saw was these aggregates of proteins. These turned out to be amyloid plaques, which aggregate between the cells, and tau that tangles within the cell. And another pathological hallmark is that as a result, severe atrophy turns up in the brain. So here you can see an Alzheimer's brain post-mortem compared to a control. And this is why you lose all your cognitive abilities. So now in the last, well, I would say 30 years of research, we have a bit of a picture of how all these different pathologies kind of lead to dementia. So what we know now is that when your thinking abilities are still normal, there's already aggregation of amyloid into plaques in the brain. And then it can take up about 10 to 5 years to show evidence for tau tangles. You see atrophy, and atrophy that really most closely corresponds with the decline in cognitive abilities. And then at one point you'll have so much atrophy, you'll have developed dementia. So this is an important point to realize, that dementia is really the end stage of Alzheimer's disease. And it's a very long disease process. So why do we need machine learning in Alzheimer's disease? Well, there's a couple of difficulties. So like any brain disease, it's really complex. There's lots of different factors that are influencing the disease, but also the manifestation, the deterioration of your thinking skills that is complex. And we now have biomarkers for the key pathologies in Alzheimer's, like amyloid, tau, and atrophy. And thanks to these biomarkers, we now know that depending on your age, many people with normal cognition show already evidence for Alzheimer's pathology, and this puts them at a very high risk to develop dementia in the future. But on the flip side, the clinical dementia syndrome of Alzheimer's disease does not always accurately reflect the underlying pathology. It turns out that up to 30% of the patients with a clinical label of Alzheimer's do not have the pathology. So if you are developing drugs that would remove amyloid, you can imagine that that won't work if you don't have amyloid pathology. And what these two extreme cases reflect is that based on the symptoms only, it's just not possible to accurately identify the underlying pathology and where you are in your disease course. And so this is where machine learning might help, because we need tools to more accurately diagnose and prognose individuals in their disease timeline. And with machine learning, we might be able to discover new markers that are more precise. Yeah, but this is immediately also a problem because there's so much data, and Alzheimer's disease itself is also heterogeneous. So that's another level of complexity. So if I say Alzheimer's disease, I mean that people have pathological evidence for amyloid and tau aggregation, and in living patients, we approximate this pathology with biomarkers measured in the cerebral spinal fluid or with PET scans. And what we now know from all these studies that have been done so far is that the same pathology can be related to different genetic causes and different molecular processes. The same pathology can result in different clinical pictures. So we have patients manifesting with memory problems, but there's also people with relatively spared memory. There's lots of differences between people and their age of onset. Some people decline very fast and others stay stable for a long time. What am I missing here? Oh, atrophy. We see differences in the precise anatomical locations where atrophy is taking place. So all these people have Alzheimer's disease, but there's a lot of heterogeneity, and that makes things very difficult. Keeping this complexity in mind, and then machine learning, which is basically just algorithms that use statistical models to learn from the data. And you can do that in different ways. So you have supervised learning, semi-supervised learning, and unsupervised learning. And I'll just cluster the supervised learning and semi-supervised learning together. And what you're trying to do here is that you know the labels of the people. So, for example, you have control individuals and Alzheimer's patients, and you have a lot of data on the other side of your model. And then the goal is to figure out which patterns in the data can best separate your controls in your cases. And this is a great advantage of these supervised learning techniques, because you can discover new associations of certain clinical or biological variables that go together with your disease. But of course, there's also a big caveat. If your control and your disease label are inaccurate, your model will always be wrong. And these types of models will not lead to an improvement of your labels. So this is something that's important to keep in mind. And I'd like to give an example of this. And I'm going to use cerebral spinal fluid proteomics because this is one of the modalities that I really like myself. And here you see an example of a table of one of the first proteomic studies. And so what they did was that they identified for 142 proteins, 300 fragments. So protein can be a very long chain of amino acids, and you can break it up, and then the little parts of the protein will be the peptides. What this group did, which is pretty neat, is that they measured this in a very large cohort from America. It's called the ETNI study, and the whole raison d'être of ETNI is to collect a lot of data and share it with the world. And so they measured the data, and of course they did analysis, and they compared these protein fragments in the cerebral spinal fluid between controls and Alzheimer's disease cases. But the labels were based on the clinical manifestation. So no biomarkers for underlying Alzheimer's pathology were taken into account. And you might remember that I just told you that some people who are considered to be controlled just based on their cognition, they do have Alzheimer's disease. And some Alzheimer's disease patients do not have amyloid pathology. So this study did not take that into account. And from all these peptides that they tested, well, you can see it in the table yourself, there were only 11 associations with Alzheimer's. So some of these protein fragments went up in the concentration, other went down, but this was less than 5%. And this is not the only study that showed little changes in proteomics data. Actually, most of the early proteomics work only found very small differences between groups. But because this was measured in ethne, we were able to see what would happen if you would improve on the labels. So here, what we did was also to compare Alzheimer's disease with controls, and you can already see that there's, with the lollipops going up and down in the end, that this is more than 11 proteins. And what we did with improving the labels was to make sure that the controls had normal CSF biomarkers and Alzheimer's disease had at least abnormal amyloid. And then we further refined the label within Alzheimer's disease because we knew from the literature that these CSF biomarkers for tau not always pick up the tangles. So about 30% of people who have tangles pathologically in the brain, they have normal tau levels. So what we did was to create these AD subgroups based on normal or abnormal tau. And this is what we found, a huge group of more proteins that showed exactly opposite relationships in the CSF of the protein concentrations. So that means that how these proteins alter in the disease depends on on the CSF Alzheimer's biomarker profile. And if you put these people together as if they would be one group, then you miss a lot of information. Almost 80% of the proteins are missed when you're not taking into account these CSF tau levels. Right. And what you should always do with statistics, whether it's advanced machine learning or simpler, is trying to see whether your results replicate, especially if you have these unexpected nonlinear relationships. And so we did that in an independent cohort that measured even more proteins, and you can see that there's the same pattern there. Right, so take home message, wrong label, and you miss relevant stuff. Yeah, so I started telling you that there's 3 types of machine learning, and the last one is unsupervised learning. And here we let the algorithm decide for itself what the labels in your data should be. And this is really exciting because you really can discover new stuff. But this is immediately also the flip side of the coin, because when you're discovering new stuff, usually there's no gold standard. And the beauty of these unsupervised learning techniques is that you can always find the results and you can always find subgroups in your data. But are they useful? Are they meaningful? So I'd like to take you through some very, very small subset of techniques that you could try out for yourself as well. But I'd like to focus also on the positive and the negative things. Yeah, and I use unsupervised learning here interchangeably with cluster algorithms because I'm interested in clustering. And what you do with these algorithms is that you assume that in your data, there's patterns present that reflect subgroups of individuals that are more similar to each other than to other individuals. And one of the simplest ways to discover these subgroups is by using Gaussian mixture modeling. So if you have a variable and you have the distribution of its values, if it's bell-shaped, then we say it's a Gaussian distribution or a normal distribution. And what is pretty cool with amyloid biomarkers, and it doesn't matter whether you look into the CSF or on PET, what you usually see is that there's two of these Gaussians together. And this is something that you can uncover and also model with Gaussian mixture modeling, the multiple normal distributions. And what it means is that this is reflecting that there's different populations in your sample. So for amyloid, we would have a normal population, an abnormal population. But the only way to detect it is that they need to be included in the data. So if we look at the example figures here on the left, if you would if you would have a population for some reason that only included people from the blue or from the red, then you would not see two distributions. There's a bit, so that was based on one biomarker, but if you talk about proteomics, then you have lots of biomarkers, and it doesn't need to be proteomics. You can also think about MRI techniques where you have voxels, or PET that also has voxels, and all these ideas can be applied to whatever types of data that you have. These are two examples of clustering techniques that can combine different variables together. So on the left, you see one of the first Alzheimer's clustering attempts using only tau and amyloid and p-tau. And there was already evidence that there might be 3 subgroups of amyloid and tau in Alzheimer's. And then to the right, you see a more recent study where people looked into gene transcripts from brain tissue in Alzheimer's disease and in controls. And there they used a different technique called weighted sample gene network analysis. So what you basically do is that you compute correlations of gene transcripts across individuals, and then you can create a network and find sub-modules in your network. So these are 2 different techniques. The k-means can deal with nonlinear interactions, which might be a bit obscured in the weighted sample gene network analysis because it's depending on linear correlations. So if it's nonlinear, you may miss it. And another thing about these 2 is that, well, you can have a lot of variables, and you can ask a question, which of these variables, for example, proteins, show similar changes with a disease. So what you're doing then is that you cluster the proteins together, and then the underlying question that you're trying to answer is that there might be biological processes that change in a similar way in the disease. On the other hand, you could say, well, I want to know whether individual patients look more similar and if we can identify subgroups. And so these are closely related, but slightly different questions. And these types of clustering methods can only answer one of those questions. And so this brings me to another clustering algorithm, which is called non-negative matrix factorization. And I'm not going into the details, but what is pretty neat about this algorithm is that it can cluster both the proteins or any other feature that you have, and individuals at the same time. So how the people are grouped together informs how proteins are grouped together and also back and forth. And this is particularly interesting because if you're investigating patients, in the end, what you want is a tool to better diagnose, for example, a patient. So this is some of the work that we did on cerebral spinal fluid proteomics. And here you see the same data that I showed you a bit earlier with the lollipops going up and down. And that already indicated that there might be 2 subgroups. But with this algorithm, we discovered that there's actually 3 subgroups in the CSF. So in this heat map, every row is a protein, and then the columns represent the average protein level for individuals with that subtype. And red means that you have higher concentrations than controls, and blue is lower. So you can see that we have a very opposite relationship for most of the proteins for subtype 1 and 3. And then there's subtype 2, and that's a new discovered subtype, and that shows a different combination of these proteins. And this is highlighting again also another neat thing of non-negative matrix factorization algorithms, that it is sensitive for nonlinear relationships. But if you were keeping attention, then you can say, yeah, but you always have a solution with your clustering, and what is your gold standard? So the gold standard is indeed absent here. But one way to figure out whether your result is just a chance finding or not is to replicate it. So this is what we did in the independent AMIF cohort. And this cohort was a bit bigger in terms of the number of patients included, but also in a number of proteins that were detected. And what you can see here just by eye is that these patterns are really similar. And because we had more proteins, we could also better interpret the third subtype, because it included proteins that were not measured in EDNI, and these were blood-brain barrier dysfunction proteins. But if you're interested in all the details, I'll refer you to the paper. Right, but you can see if you have more proteins, then you can discover more processes. So we thought maybe if we increase sample size even more and detect more proteins, maybe we can discover more subtypes. And now we found 5. So we replicated the 3 I just showed you and discovered 2 new ones. So this is another thing to keep in mind. If you're interested in refining this biological entity, the larger the sample size, the better, because if there are subtypes present in a disease, then you need to have a big enough sample size to be able to find these subgroups of people who are more similar to each other than others. Yeah, so I'll wrap it up quickly because I think I'm going over time a little bit. If you, I have a spaghetti elephant here, but you can ask questions about that, what I mean. What I hope that you picked up from this webinar is that there's this distinction of supervised learning and unsupervised learning. And be aware that with supervised learning, you can never do better than what the quality of your label. But if it's correct, you can find new patterns. And unsupervised has this more negative quality that you can always find a solution. And I hope that I've showed you now how you can be aware of whether or not your results make any sense. All right, so with that, I'd like to end and acknowledge everybody who's involved in this type of work. And I'll stop sharing my screen so you can ask questions.
Thank you very much, Patty. That was a very nice overview of all the work that you've been doing on CSF proteomics. So I have a couple of questions here in the Q&A. So the first one is from Professor Guus Smit from the VU University. He asks if there's a minimum of group of cohort or sample size for the unsupervised machine learning. Is there a threshold of patients that you need to include?
Yeah, that's a good question. If you don't know a priori how many subtypes there are, this is a very difficult thing to estimate beforehand. And this is another thing with clustering. You have as many clusters as that you have individuals.
Yeah.
Because those are the most unique. But I would say that and if you have a very strong phenotype, you might not need a lot of people. And that's kind of most evidence from the Gaussian mixture modeling. So if you would have 10 people all the way over here and 10 people over there, you would see these two different groups. So it depends a little bit on the complexity of the data and what you know about the biological variability. But I would say larger is always better.
How many CSF samples were you able to include for your last slide where you identified 5 subtypes?
Yeah, there we had 419 patients.
Yeah,
and that was a doubling of the amount of patients we had in the previous study that were about 200 patients. So possibly if we could increase the sample size to 1,000, you could further refine it. Also, if you have more rare genetic causes in your patient sample, you need to kind of increase sample as well to be able to have them in your data.
Yeah, makes sense. Okay, next question from Odile van der Heuvel. I think this is a very relevant question. So are these 3 or 5 clusters different in clinical profile or disease progression? So when you take the CSF, are they in the beginning of the disease stage or end stage?
Yeah, it's a very good question. And also these are different aspects. So what we see is that people in the 5 subtype cluster definition, they differ in their atrophy patterns and in how fast they progress in the disease. Also, we see differences in survival rates. But if you look at the baseline clinical characteristics, then we see that these subtypes are already present in a very early clinical stage. That is that amyloid is abnormal and cognition is still normal. And what we've explored in this new data and what we want to further study in the future is that this seems to indicate that the proteomic profiles are reflecting genetic risk because it's present so early in the disease.
Right. And for the sporadic donors without a genetic background, could they theoretically move from one subtype to another as the disease progresses with different markers that you find in the CSF?
Well, this is something that we studied earlier with repeated proteomics, and what you see is that these subtypes are quite stable. We can even see 2 of these subtypes in control individuals who still have normal cognition and normal amyloid markers. So 2 of these 3 subtypes we identified, and those individuals also had repeated amyloid and tau measures. And only people with attenuated Alzheimer's subtype profile, they showed that their amyloid and or tau values changed. And then we also discovered a 3rd subtype there, but there's no amyloid or tau changes over there. So maybe that's the real normal.
Okay.
Yeah, so that's also kind of suggesting that these different molecular processes are really a trait.
Right. Yeah, great, thanks. One final question from Professor Paul Lucassen. If an increasing number of CSF sample is the way forward, will we even have more subtypes? So now you increase from 3 to 5. Do you expect more in a follow-up study?
Yeah. Yeah, no,
Okay.
I really do think so, because there's now about 85 genetic variants that have been associated with sporadic AD, and so these all have very small effect sizes. It means that not every patient has the same genetic risk factors or the combination of it. So definitely, I think if we increase this even more, then we'll further be able to refine it. But there'll be at one point a limit to the number of subtypes, probably. I'm not sure. It's an interesting question to also look into that more mathematically.
Great. So I think our time is up. We have to move on to the next speaker. There are still some questions coming in, but we can answer them in our Q&A session. Thank you, Betty.
Thank you.
All right. Thank you, Betty. That was great. So now it is my pleasure to introduce you to Arman Eshaghi, who is a colleague of mine and an NIHR Advanced Fellow at UCL, University College London in the UK. His research is focused on developing advanced AI models using brain imaging and also electronic health records, which is, I think, quite a new way of doing things in the MS field, from hospitals in the UK and Canada, and also very soon in Amsterdam. He obtained his MD in 2013 from the Tehran University of Medical Sciences in Iran, and then moved to the UK, where he got his PhD in computational neuroscience at UCL in 2018. He is quite a rising star in the field, I must say, and recently got quite a few prestigious grants, like the Young Investigator Award at Tijms, the Jacqueline Dupre Grant in 2011, an Tijms Magnums Fellowship, and an MSIF McDonald Fellowship as well. He's also on the editorial board of Neurology, and it is my great pleasure that he's here today to talk to you about machine learning in MS. So thank you very much, Arman, and go ahead.
Thank you very much, Melo, for the introduction. It's a pleasure and honor, everyone, to be here. And good morning, good afternoon, depending on where you are. During the next 20 minutes, I'm going to basically build up on what Betty showed us and getting into a slightly different field of multiple sclerosis. The intention of the talk is really not to focus on any specific disease, although I'm an expert in MS, but my hope is that by the end of it, you'll be able to get ideas to apply it in your own field of research, and that doesn't necessarily need to be MS. And therefore, I'll give you lots of examples that are not related to even neurology and other disorders. Specifically, look at the concepts on stratification and what that means and what machine learning can do for us. Classification, Betty touched on that a lot. I'll try to approach it from a different perspective and look at the AI or machine learning in how it can be used as a crystal ball to tell us about the future. What the exciting area, as Melo mentioned, is the new ways we can now start to look at the data we have and the data silos we have across the hospitals, and the way AI can enable new ways of looking at the data. We also look at the future, how future may look like in the next, say, 5 to 10 years. When we look at the specifically neurological disorders, when we look at how diseases are classified, they are almost always based on symptoms, very specifically in multiple sclerosis, but also and even in Alzheimer's disease and other neurodegenerative disorders. This is really historical. When you look at the histories of these disorders, they have been usually diagnosed and named by neurologists who look at the symptoms. The problem with symptoms is that similar symptoms may be the result of different underlying pathophysiologies. And for that, what we see on the right, there is a paradigm shift, again, not just in MS, but this is about very specifically on Alzheimer's disease. We are looking at the ways of using the underlying pathophysiology to classify different neurological disorders. This is the ATN, amyloid, tau, and neurodegeneration axis and classification proposed for Alzheimer's disease in a research setting. So instead of looking at symptoms, looking at the dementia, we could start, at least in a research setting, to use the underlying changes of disease to classify the disorders. And why is that so? The reason for that is the move towards the group level, empirical medicine, towards the individual level, classification and staging of disorders. So when we talk about classification, staging also becomes a fundamental issue, and that is because with at least in neurodegenerative disorders that they may take decades to manifest and to worsen, we need a way of objectively staging patients. So that's basically what I called initially stratification. So we have different concepts of stratification, staging, and classification, but they come together to tell us more about an individual as opposed to the group or a disease at a group level. So I get in my next slide, get to how, again, MS is an exemplar, is just a template for you to think about the next few decades of medicine that we're going to shift the paradigm and looking at different ways of classifying disorders. So this is a classification we have had since 1996, led by Fred Lublin. And what the Fred Lublin and the colleagues, they looked at the symptoms, how they evolved to classify MS, including relapsing remitting patients who have wax and waning of symptoms. Most of these patients go on to develop within 10 to 20 years a secondary progressive course. We also have about 10% of patients who have a gradual worsening since the very beginning of the disease, called primary progressive MS. We see over time, so this was 1996, we're looking at 2013, over time, there is a move, as I showed, there is a move towards trying to look at the underlying pathophysiology of disease. So we're looking at activity of the disease in MS, for example, using MRI, and progression using MRI. So it's actually for the first time, we're looking at MRI and other biomarkers to move away from symptom-based classification towards a more biological understanding of the disease. And why is that necessary? As I mentioned, we have empirical medicine. We have had the kinds of Alzheimer's or Charcot and other people who looked at individual diseases instead of individual patients. They came up with classifications. We are pushing the envelope here as you look, we're moving towards the stratified medicine. Hopefully one day we should get to this personalized or individualized medicine. But to get there, as you see, the stratified medicine is the, I'd say the next, or now perhaps the next few decades, we will be here until we get to the point that we can actually look at individual patients. So looking at the staging of how we can use machine learning and AI to stage patients before getting to classification of disease, we looked at the data in the Magnims cohort a few years ago. We used a machine learning model called event-based model. And what this machine learning model does, it looks at the staging using solely on MRI. In the clinical practice, we use EDSS, disability, and other scores come from clinical examinations to score patients. Now we can use MRI to, for example, looking at the brain shrinkage, brain atrophy, to look at the staging of different MS subtypes. What is really striking from this slide is that irrespective of the clinical MS type, you see ABC, the MS clinical subtypes, they're actually more similar than different when you look at the progression. This basically tells us that as I said, the symptom-based way of classifying disorders is probably doesn't tell us too much about the underlying disease pathology. And as you see on the bottom right, patients, their staging obviously goes up in over time, and patients with progressive MS, they also were more likely to have a faster staging over time. But the problem, as I said, we thought about this problem, we realized that we needed a new framework to be able to stage patients. So first, we need to have a classes of patients, not to try to reclassify or redefine MS based on the changes on MRI. And I keep saying MRI, not just because, MRI is one of the many different biomarkers. Betty mentioned CSF. These are really other biomarkers and really important. I use MRI because it's widely available. It's at least, the low-hanging fruit in the field. You can get the large data sets using MRI, at least in MS and neurodegenerative disorders. So here we looked at the same problem I showed you in the previous slide, but with a new lens. We said, okay, we're not going to make any assumption about the subtypes of MS. We leave it to the data to speak for itself. and then try to understand the subtypes, and then use that new subtypes, also stage those subtypes. What we found, we looked at the huge number of patients, about 9,000 patients. These were collected in Montreal Neurological Institute, where I also work with Doug Arnold and others. We looked at the progression of MRI abnormalities, and the three subtypes emerged that we call them based on the pattern of MRI abnormality: patients with lesion-led, white matter-led, and cortex-led. Patients, for example, with cortex-led, they had a predominant shrinkage in the brain as opposed to a predominant lesion pattern in the lesion-led subtype. Why is it important? Well, it could be important when you look at the treatment effects. We see on the right, when we looked at the these are looking at randomized controlled trials in patients who in MS, we do have treatments that actually work sometimes. And we see on the right that patients who had a lesion lead, they had about 80% increased when we looked at the rate of disability worsening. So that's the way how we define treatment effect. This is probably known in MS because these patients are active, they're more likely to respond to the disease. But what is striking is looking at patients who may be not necessarily benefiting with the treatments, and try to develop the next generation of treatments for those patients. When we looked over time in these three groups, we also see that patients with lesion-led, with both the training data set, you see the number of patients in the bottom, and also in the external validation data set, again, we looked at the randomized controlled trials. Patients with lesion-led, they had a worse prognosis as defined by what we call EDSS progression. This is disability progression. This is just a way of validation. As Betty mentioned, there's no gold standard when you come up with these data-driven subtypes. You need to have clinical measures, in my opinion, to be able to validate in a way those outcomes. And there is I can tell you, there's, the next classification of MS, the next 3, 4, 5 years is not going to be based on symptoms. Hopefully, we're going to see more biological ways of classifying MS. There is an active work group led by the National MS Society in the United States that they're looking at these problems. MRI is just part of the puzzle. There are lots of other biomarkers can be used. There are lots of question marks whether all these variables we see, they can be actually used in clinical practice. There are lots of aspects to it. I'll discuss later about how we can translate these sorts of models into the clinical practice. But at least for the next step, or at least next iteration of this classification, what we would expect, I don't think we would expect to see machine learning to be used in classification, not probably for the next 10, 20 years in clinical practice. But at least we would see mechanism-driven subtypes, hopefully, by having biomarkers incorporated. So watch this space. The next few years, hopefully, we see more of these kinds of classification. So one question, as I mentioned at the beginning of my slides, I'm not really going to focus on one disease. And we can, as neuroscientists, as neurologists and clinicians, we can look at cheat a bit and look at other fields, see how they have looked at this problem, especially the field of cancer. So this is one question we were asked. So how can these classes of disease, data-driven classes, be used in a, for example, clinical trial setting? So this is a study published in Nature. It's done in the UK, in Birmingham. Is a clinical trial. It's called an umbrella clinical trial, where patients with lung cancer, they go through standard of care therapy on the treatments they have. And if the treatments, they don't work, they go through high throughput genetic screening. And based on the mutations, they receive different treatments. So this works like an umbrella. Think of an umbrella with different branches when it opens up, and they receive different treatments. You see on the right, different classes of drugs. And these drugs, they are targeted towards the mutation. So, this is the move, is in practice how stratified medicine can work. And again, this is far away from a clinical practice, this clinical trials. These are two different worlds. But still, we see the next few decades how it may look like for neurology, how we can use these kinds of ways of classifying patients, subtyping patients, and then use that to develop new treatments and even understand the disease better. So one other thing that I'll mention is the prognosis. So we looked at the classification. Prognosis is the other area that, in my opinion, at the moment is really done in MS in the clinic by simply a discussion between a patient and a clinician. So we can certainly do better. We can objectify or move towards objective ways of at least informing that discussion. And I feel the prognostic models, AI models that can integrate different bits of the data we get from patients, they can be used to predict the outcomes and also the risk of those outcomes, the time it takes for someone, for example, to become wheelchair bound. I'll give you quick examples and brief examples, and then I'll move on towards more on clinical side of things. So how can we use machine learning and AI for integrating data in clinical trials, for example? So this is a study that's not yet published. We looked at the deep learning models that it can get clinical data. By clinical data, I mean disability and the very basic demographic data that are widely available, and also MRI data, which included, for example, brain volumes. We looked at the normal, the white matter T1, T2 ratio, and also lesion volumes to predict the patients who are at high risk of disability, medium risk, and low risk. And also when you integrate this, and what we looked at, as you see on the vertical axis, is the confirmed disability progression. This is the outcome used by FDA to approve treatments. So the one question, how these AI models can be used in, at least in clinical trials to start with, do we have this new concept of prognostic enrichment, that is the way we can use AI machine learning models to screen patients into the next generation of clinical trials. So here we're looking at in silico, these are simulation of clinical trials, looking at phase 3. So phase 3 trials are where you look at the drug development towards the end of approval by FDA. When we look at if we can enrich trials by patients who are at higher risk of developing disability progression, you see that if when you use the AI models, we get about 10-15% increase in the statistical power. And if we want to think about, well, how does this translate to the real life? This translates about a reduction about 4 or 5 months in patient recruitment. It may or may not be practical yet and economical yet to be used, but it at least shows us what can the next few years look like in integrating AI machine learning into the running clinical trials. So I'm just getting to the second half, and towards the end of my talk, I wanted to speak about how these advances, the technology, can be used for the benefit of patients. I'll never forget whenever I worked at this, at the end, we want to convert this from an academic exercise to a life-changing technology for patients. So we look at the hospital data, which is the area I'm funded to work on the next few years. Clearly, the real-world data is messy in a way. The data quality keeps changing over the years. You cannot control it. It's not research data. Data constantly changes. And of course, we have privacy issues. I'm going to touch on these different aspects very briefly the next few slides. The first one is how AI machine learning can unleash the data we already have amassed in our hospitals. So we're talking about tens of thousands of patients' data available, again, is in the UK or in Netherlands or any other country in the world. You can get access to large data sets. The problem is these data sets are really not useful the way they are for clinical use, or at least for clinical research to start with. So what I'm showing you on the left is the latest advance, in my opinion, in the field. This paper led by Junio Iglesias in MGH, in Mass General Hospital. He and his team have developed a way of converting any sort of, I would say, routine care quality data. We looked at the T1, T2, and FLAIR MRI that are usually not used for looking at brain volumes. We have this technology called SynthSR that converts those lower quality data into the 3D T1 weighted MRI, which can later be used to, for example, quantify biomarkers. This is quite striking because suddenly it unleashes a lot of dormant data and can be used, to answer even very basic research questions. On the right is a work done by the group in Amsterdam by Piet. And what he looked at the MRI data that we have, again, clinical data, T1, T2 MRI, to generate advanced MRI. So he looked at the double inversion recovery, which he uses or looks at the cortical lesions, as we see here, again, without getting into details on why this is important. It enables a potential new line of research where you can generate and ask even basic science questions using clinical data. So you see that the translation or even reverse translation enabled by the new AI technologies. We are working on similar models, especially the ones on the left in the MS. We're looking at ways to unleash these huge data sets from both electronic health records and also the imaging data. I looked at the privacy concerns. So we are looking at ways of looking at the data that precludes the need to share data. Patient data is extremely sensitive. We need large data sets to look at some of these problems. The problem is we cannot share data between different centers. We are looking at the number C and C. So the way we do the research at the moment is A and B, basically. Mostly you store your data in one server, then you start to run your experiments and you start to try to answer questions. What we're looking at is to have different models trained separately. We're looking at the Nature paper that looked at the prediction of leukemia. Again, it's a completely different field, but the idea was to predict leukemia in the future. What the swarm learning, what it enables is that if you train these models and you look at the accuracy of these models, what you see on the right, you can always improve the accuracy and even to get to the point that the central way of sharing data would do. So basically, you really don't need to share data anymore, in my opinion, if we can design these models using these newer technologies. So I just want to look how we can translate this to have better evidence for the future. The problem we are facing at the moment, in my opinion, is that we are really in really early days of development of AI models. We do almost everything offline. The next generation of models is to be able to do them prospectively, to run randomized controlled trials, the way you run it for treatments. And this may take decades. So how is it done? We're looking at an example from the cardiology field where patients are referred using basic technologies like an ECG that has been done for decades. And then they need to be screened for more expensive technologies to be screened for heart failure. So we're looking at an AI technology that interprets ECG and tells the clinician if the patient is more likely to develop heart failure. And we see that this is a randomized pragmatic control trial. Doctors who had access, what you see on the left, who had access to AI results, they were more likely to have the correct diagnosis and detect heart failure earlier. And you know, the important point, which I always say, is that we're looking at the AI plus doctors rather than comparing, for example, doctor versus machine learning or any kind of that, which I think is an ill-posed question. So we can think about this, how we can use the same ideas in the future in neurology and run prospective clinical trials. Hopefully one day I'll present a potential clinical trial in MS and looking at these kinds of prospective studies. So again, AI and machine learning, we are looking at ways of having a continuous measurement from patients. You don't necessarily need machine learning to be able to get patient data. On the left is an iPhone app, and on the right is a NeuroKeys. Again, this is led by Joep Kleestein and others in the group in Amsterdam. Again, you don't need machine learning necessarily, but machine learning enables you to look at this large data set in a continuous manner and try to, for example, have an alert system to predict the future. These are the research in early days. It's unclear how this might be one day translated. But we look at again, another disorder, looking at the Duchenne muscular dystrophy, how AI can be used with this wearable technology to help patients is to develop new ways of looking at disease, to develop biomarkers that are more sensitive, and they can be done in a real life setting, in a patient-centric setting. So these are patients who do assessments based on, these are mostly children with this Duchenne muscular dystrophy, and they have this wearable, but they do basically daily activities. They don't need to go through clinical assessments. And this work was done at UCL at GOSH, as Great Ormond Street Hospital. And what we're looking at is this is a 6-minute walk test. Is that the way we look at traditionally in this Duchenne muscular dystrophy, we look at the worsening of disease using this biomarker. This is already out there. It's based on clinical exams. It's very noisy, as you see on the left. It should go up because patients get worse over time, but because they can move less than the biomarker is not very useful. But what you see on the right, we can use these wearable technologies as expected. As patients age, as their disease gets worse, this biomarker goes up. And this, when you look at it, this is less noisy. And also it helps us to design clinical trials with less number of patients with more power. This is my last slide, and then I'll stop. So I just, in the past 20 minutes, I tried to say why we need machine learning. Machine learning is just a tool. It enables basic fundamental questions that hopefully will change the paradigm for patients, to benefit patients. The idea is to have AI machine learning as decision aid systems one day, which we don't have yet, but I think it takes decades to get there. We might be able to use AI machine learning to have new outcomes. And as I said, the path to clinical translation, similar to developing treatments, is very long, it's decades. That needs research, that needs investment from funders, and that needs really deep thinking and not really rushing to conclusions. At the end, I want to thank the UK government. They fund my research, and myself, my research group, which I have recently started, and my collaborators at UCL, at the Montreal Neurological Institute, and also you for your attention.
Thank you very much, Arman. That was really great. And also highlights the evolution I think the MS field, but also neurology and perhaps medicine in general is currently in. We have a question here from Nikunj Davda about the different groups that you created with cortex-led, lesion-led, and neural pairing-led. And the question is, did you also look at more advanced aspects of the treatment? So when it was given, when it was begun, and also the intensity of the treatments?
Yeah, so thanks for the question. This was done in a setting of a randomized controlled trial. So, if you ask me when the treatment was started, it was started exactly when patients were classified. So we had the time 0 at the time that patients were classified and were given the treatment. We have lots of different treatments for MS, and in that data set, in high efficacy treatments, obviously, we could see more treatment effects, specifically in the patients with lesion-led. But in other groups, we couldn't see, at least at the group level, we couldn't see any treatment effect, irrespective of treatment.
Then I was also wondering, you spoke also about that we need quite a lot of years still until we are in clinical practice with these kinds of techniques. But what do you think is the kind of information that neurologists for example, in the MS field are really looking for that AI could provide? So how would this work in clinical practice if they have a patient in front of them?
Most of the things we do in clinical practice is very much subjective. So I think anything that can help us move towards from subjective discussion toward anything objective would be helpful. I would think of looking at the risk of getting worse, the probability of responding to specific treatments, that would help. But on a more generic basis, anything that can benefit patients can be useful. But at the end of the day, I think how this could be implemented and how it could help, that in itself requires research. So how is it going to be part of the conversation? Is it just going to be processing of scans? And whatever that is, that in itself, in my opinion, requires evidence generation. So say, for example, we need to run a trial, see if we add, say, numbers coming out of machine learning to the discussion, can this benefit patients? And that can be quantified. And if so, then that would be the way to integrate it.
Yeah, I agree. There's another question here. Can AI or machine learning better handle multi-center imaging data harmonization and how effectively could it be achieved? Thank you, from Yong Lee.
Short answer, yes. Initially, when I say, early on, maybe 2015, 16, 17, these AI models are extremely fragile. So once you go from one center to another, it basically breaks apart. I'd say I showed you one slide on the data harmonization. I didn't mention data harmonization, but the idea of SynthSR, for example, was data harmonization. We are, I think, on the cusp of something really big where we can use the newer technologies where they are quite robust to multicentric data. They can be used for data harmonization. And in a way, maybe we don't even need data harmonization, in a way that if the models are trained in a multi-center setting, that they should be able to help us or perform in different settings, or even data that change, and they may even have different quality and so on.
Mm-hmm. And it was a question that Betty also got. I think it's quite interesting. How can we, with such advanced techniques, how can we disentangle if the results are true phenotypes of patients or different stages of the same disease?
Yeah, so I don't have any answer for that. So we looked at the subtypes we had within 2 to 3 years of clinical trial, and I don't think that's enough to answer your excellent question. You didn't see switch between subtypes within 2-3 years. Mm-hmm. But I think that requires at least, MS or even any sort of neurodegenerative disorder may go on for decades. I feel we need to look over a longer period of time to be able to answer that question. Is it just staging, or is it subtyping?
Yeah, I agree. So what I think we'll do now is invite Betty and Anke also back. And if you have additional questions also for Betty or for anybody in particular, do post them in the Q&A. I thought of one question maybe for both of you. One aim of TN2 is also to look at valorization towards industry and back again. So what do you think in this field is the role of industry, and how can we come together to, in the end, to help patients, of course?
Can I?
Yeah.
Well, those biological subtypes that we see, they show very distinct differences in certain processes for which drugs already exist. But the treatments, the trials, they got stopped because there were side effects, or maybe it looked like there wouldn't be any effect. And one of these is BACE. So BACE 1 is secretase that cuts the amyloid protein and plays a role in amyloid metabolism. And what we see is that these subtypes, they can show really high amyloid or very lower than control levels. So if you inhibit that when it's already below normal, then for sure it will give more side effects. So this is how industry might help us, that we could reanalyze such trial samples and figure out why some trials went wrong and learn from it.
Yeah, I agree. Arman, what's your take?
Yeah, so thanks, Betty. I fully agree. I think it's exactly what I wanted to say. But I think I can add to that. When we look at the technological readiness level of the ideas we have, most of it is, a lot of it is academic ideas, which is blue skies research type of thing, extremely important. But to translate this to or scale it up to a randomized control trial is like a drug. Once you develop it, you have some proof of principle, you publish your Nature paper, then the next level is to have a very large clinical trial. That is really not possible in the academic setting. So we look at I would say it's the success story in MS, in the pharma industry, running phase 2, phase 3 trials, and even late stage phase 1 trials. I think that's where we can get these AI technologies, could be subtype classification, and run those. I don't think pharma should do that, but I think medical device companies are well placed to do that. So that's one side of the story on the funding part. And the other side, how they can give it back to the community. I would think about the ways of the amassing data and reusing that data. And a lot of my research I've done is thanks to the industry, because huge data sets are becoming available. And I think getting access to those kind of data in this new setting would be extremely helpful.
Anke, go ahead.
Yeah, great. So we still had a question open from Betty Session as well, and I think this also applies to Arman's data, which is what is the accuracy of subtype classification at the individual level? So basically, it relates to Arman, where when the patient comes to the clinic, can you actually make a diagnosis? Can you make the subtype classification already when the patient comes to the neurologist?
Yeah, so thanks, Anke. You can't, I mean, from a technical point of view, it works at the individual level, but when I say technical level, when you want to translate to the clinical practice, not practice, research, I would say, I don't think we are even close to having this in practice. In clinical research, the question completely changes. So I don't think this would help any patients at individual level, despite the fact that we can provide a subtype at the individual level. And why is that so? Because we are not accurate enough to be able to predict. I'm sure that there are more than 3 subtypes that can explain why your MS is getting better or worse. So that's just one thing to start with. And the other side is that when we looked at the treatment effect is so variable that is not yet useful for us to be able to use those subtypes. There's another problem, is that how we define benefit for patients, at least in the setting, a clinical setting for multiple sclerosis. We use a very crude measure, which is accepted by almost everyone, including FDA, which is called disability, or EDSS, I call it. But basically, disability is extremely crude. So that's the other problem. So unless you have a good measure that can tell us, look, this patient is getting better or worse, we wouldn't be able to have a subtype or an AI model that can. So it's like, it's a two-way street of how we can have an individual level prediction.
Thanks.
Yeah, and I like to, so those are really good points, that from discovery to clinical practice, there's, big gap. And before 2020, nobody even knew of the existence of these subtypes. So you need to go through several steps. And we can classify them with really high accuracy, up to 90%, but we only have limited data yet. So we need to replicate and bigger samples, and then we move towards developing panels. And then hopefully at one point we can also show that it might be helpful as stratification for treatments, but there's many steps that need to be taken.
So also what made me think was the examples from other fields, like from cardiology and these things. One of the difficulties that I always encounter, and I think all of you as well, is that a lot of insights come from these different disorders maybe 10 years later. So how do you think we can promote sort of accelerating the AI field by coming together? How are we supposed to do that?
Yeah, Betty, you can go ahead.
Yeah, I said you're asking us.
Yes, of course.
Yeah. So I think that something like TN2 is really important, because you have like the technical developers on the one hand, and you have the clinical people on the other hand, and all types of researchers in between. So what you need is to have a good dialogue between people who are going to use it and people who are developing it. That needs to be continuing. And perhaps, Arman, you'd like to add stuff.
Yeah, I think, there's a lot of work. So, as you mentioned, rightly so, there's a lot of work that's gone into thinking about designing even, research studies for testing or developing AI models. I think probably for us as being in a different field than cancer or cardiology or whatever, is to, we can reduce the time for translation by reusing that technology or using those kinds of studies as templates. So that was actually what I was trying to say, to reduce the time for translation and developing these models. But obviously, neurology is very different from cardiology, so we also need to make a lot of modifications and so on.
And also, we don't necessarily read papers from cardiology, so I think that's also a bit of a problem. How do you find them? How do you know that they're there? Apart from Twitter and big news items, of course.
Yeah, but I do like to add that the brain is really the next level complexity. So it's not straightforward how perhaps advances in other fields might also be already applicable to what we're trying to do.
Yeah, I agree. Thank you very much, everybody. I will just share my screen quickly for this final bit of information. As we said before, we're going to have an in-person meeting next in April, on April 5th, at the Amsterdam Science Park, very close to where Anke lives, works, sorry. So the link to register for this will be in the chat. As you can see, we have 2 keynote lectures, one by Dick Swabe, one by Valentin Riedel, and a very nice program that will go live very soon as well. So thank you very much to Betty and Arman for coming, and Anke, maybe you have some final words as well.
No, I would like to all welcome you in Science Park 5th of April. So hope to see you there.
Yeah, great. Thank you very much, everybody, and see you next time.
Thank you. Bye.